session. Thank you all.
Hi, everyone. Thank you, Mir. Well, I am Alex Murillo from the Cancer Research Institute in Sao Paulo, Brazil. And now we will continue with our event. For now, we will attend a presentation of Laura Nicastro from the Imperial College of London. Laura, are you here?
Yes. Hi, I'm here.
Hi, that is a pleasure to have you here.
Yeah, I can
please
share my
feel free.
screen.
Yes, please feel free to share your presentation.
Okay. Do you all see it, or can you see the presentation?
I hear not yet.
No?
Not yet.
Ah, sorry. Probably it's better if I do like that.
Yeah, there is no problem.
I'll try now.
Now it's
working?
starting, I guess. Yeah.
Okay. Sorry, 1 second because my mind froze. I don't know why my computer froze. Can you hear?
Okay.
Yeah, I think it's not only
we are
yours.
hearing you, but
We see the black screen.
yeah, now it's black.
I think
Stop your presentation and start again, Laura.
And the thing is that my computer momentarily froze. I don't know why. Okay. Ooh.
Okay.
Um,
Things happen.
I'll repeat.
There is no problem. That's normal.
Sorry. Should be fine now, maybe. Can you?
Yeah, we can see it now. You can start.
Okay.
Thank you.
Perfect. Okay. Sorry for the inconvenience, and thank you for the introduction. I'm Laura. I'm a final year PhD candidate at the Imperial College, and today I'll present my talk, Regulation of Myocardial Contractility Mediated by Extracellular Vesicles and Studied Using Thin Myocardial Slices. So first of all, a bit of background. So the transition from a healthy to a failing heart, which is caused by excessive mechanical load on the heart undergoes substantial remodeling in terms of development of hypertrophy, electrophysiological remodeling in terms of calcium and contractility, and reorganization of overall extracellular matrix, and death of the cardiomyocyte. Between the numerous humoral and neurochemical responses that act to remodel the heart, extracellular vesicles are an important key player in the paracrine crosstalk between cardiac cells in the heart, because they are able to shuttle bioactive cargoes, for example, RNAs and microRNAs, to the different cardiac cells in the heart. And as you can see here in this schematic, they're able to send each other messages to promote or reverse remodeling. Now, the current platform that we use in cardiovascular research to study the effect of extracellular vesicles in the heart are either too simplistic, like in the case of cell culture, because there is no connection within all of the cardiac cells and the extracellular matrix. And in case of isolated cardiomyocyte, they differentiate very quickly in vitro. Ex vivo isolated heart are just not suitable to perform chronic experiments, given that they're only viable for a couple of hours, whilst in vivo animal experiments, which may seem the most suitable, are too complicated to investigate the mechanism of function and isolate the heart from the other organs, as well as considering the ethical consideration. In our lab, we have developed a platform that sits exactly in between in vitro and ex vivo isolated heart, which is called living myocardial slices. Living myocardial slices are prepared from different species, as you can see here, ranging from the rats until the human, and they're prepared by isolating a left ventricular transmural block from the free wall left ventricle of the species, And they're placed in a high precision vibratome, which slices 300 microns thick cardiac slices, which then are going to be trimmed, and some rings are going to be attached orthogonal to the direction of the fibers to recreate a model that can also be stretched. There are many advantages compared to current in vitro and in vivo models. First of all, we reduce the animal suffering, and the slices can be prepared, as we can see, from a large variety of species. But most importantly, slices can maintain the multicellularity, architecture, and electrophysiology of an adult myocardium. And because they can be cultured up to 5 days in vitro, we can perform chronic experiments on them. Moreover, living myocardial slices can recreate, we're able to recreate with them disease models in vitro, like, for example, the application of mechanical load, which is specifically what I will focus on today in my talk. So the first hypothesis that we had was that extracellular vesicles released from myocardial slices that are stretched at different mechanical loads can influence the functional properties of myocardial slices. We did it, we prepared the living myocardial slices and we stretched them at 2 different sarcomere lengths to recreate, which are 2.2 and 2.4, to recreate respectively physiological and overload model. So overload is very similar to a failing myocardium in vitro. And we placed them for 48 hours in a biomimetic culture, which contains constant electrical stimulation, oxygenation, and perfusion of the culture media. Following 48 hours, we collect and we harvest the media for exosome isolation through size exclusion chromatography. And here, in our first set of results, we can see that the slices do exhibit a different have different phenotypes in terms of contractility, as we can see in the active force graph on the left, without altering the stiffness of the slice, which is the passive force. When we quantified EVs after 48 hours from these 2 different conditions, although there was no significant difference in the distribution nor the size of these EVs, overloaded EVs had a significantly higher protein concentration compared to the physiological. And here we just have a characterization with the electron microscopy of EVs from physiological slices labeled with the CD63 tetraspanin. What we did after was that we applied both physiological and overloaded extracellular vesicles on top of physiological slices, just to see what happens, how they act on the function of the slices. And we cultured again for 48 hours in the same biomimetic culture. And what we measured is, again, the contractile parameters. What we observed, so here you can see in green is physiological EVs applied on top of physiological slices. In blue is the control, so physiological slices treated with vehicle PBS. And in red, overloaded EVs on top of physiological slices. What we noticed is that overloaded EVs did not have any effect on physiological slices, but physiological EVs were able to not only increase the active force of physiological slices, but also to reduce the passive tension and therefore the stiffness. So we then wanted to see if physiological EVs were influencing also the properties of not only physiological slices, but also overloaded myocardial slices to use as a potential treatment. We again treated slices, but this time, instead of having physiological slices, we overstretched the slices to recreate failing hearts. And we applied the ranging concentration of EVs per slice. Again, cultured for 48 hours in the biomimetic culture. And this is what we have seen from the second set of results. So we can notice here that compared to the control that you see in red, a minimum concentration of 10 to the 8 particles per slice was necessary to increase the contractility at maximum contractility, so from 25 to 30% stretch of the slice. This effect was dose dependent because also 10 to the 9 and 10 to the 10 EVs per slice were able to increase the concentration, the contractility of failing overloaded slices without any alteration in the passive force. So then we said, how do physiological EVs influence the functional properties? Do they affect any aspect of remodeling. And we started by investigating the cardiomyocyte size and the hypertrophy. So we see that even though there is a difference in the cardiomyocyte length and overall area between physiological and overloaded cardiomyocyte, application of EVs on overloaded slices did not contribute to reduce their size. So they don't affect hypertrophy aspects. Then we moved on to analyzing the fibrosis remodeling aspect, and we saw at structural level there was no significant difference between the three groups in collagen accumulation or expression of vimentin, which is proliferation of stromal cells. The same effect were seen at protein level when we quantified for collagen 1 and alpha-SMA, which stands for activation of fibroblasts, even though here we had a trend towards an increase in collagen between physiological and overloaded slices. But then when we looked at the gene level, we did see that there is an activation of the fibrosis process between physiological and overloaded slices when screening for all these profibrotic markers. And in many of the cases, like for example, in the case of TGF-beta, fibronectin, and SMAD3, EVs were able to reduce some of the expression of some of these genes. So they partially probably affect fibrosis, the EVs. After that, we wanted to investigate apoptosis, so cardiomyocyte death, by expression of PARP1, which is a downstream effector in apoptosis. And we saw that there is a significant cleavage of this protein in overloaded slices compared to the physiological, meaning that the more cells are dying in the overloaded slices. And this application of physiological EVs on overloaded slices were able to partially restore the death of the cells, which we can also see by this Western blot for BCL2, which is a pro-survival marker, which was drastically increased upon application of physiological EVs on overloaded slices. This was also confirmed at the structural level when we assessed for tunnel staining, which stains for apoptotic nuclei, and we saw that there was a significant increase in the apoptotic nuclei of overloaded slices compared to physiological, and these were restored almost to physiological level when we applied the physiological EVs on top of overloaded slices. So these physiological EVs may have also an effect on rescuing cells from apoptosis. One of other aspects of remodeling is the rarefaction of blood vessel. So we stained slices for isolectin beta 4, which is a marker for blood vessels. And when we quantified the presence of the vessels in living myocardial slices, we noticed a drastic decrease in blood vessel in overloaded slices compared to the physiological, but these again were brought to almost physiological levels when physiological EVs were applied onto overloaded slices. And when we see that at gene level, there was an significantly increased expression in the potent angiogenic factor VEGF in only in overloaded slices that were treated with physiological EVs. So then we said, how do these EVs modulate these changes that we see in contractility and remodeling? And we performed a microRNA array analysis on rat physiological and overloaded EVs. So what we observed here is that we found 2 upregulated microRNAs in physiological EVs compared to the overloaded, and 2 downregulated microRNAs. You can see here in the heat map the pattern of expression of these different microRNAs and how it's substantially different between the 2 conditions. We then decided to focus more on the overexpressed microRNAs in the physiological EVs because, as I guess many of you know, microRNAs have the main the main action of the microRNAs is to inhibit the expression of the genes. So we only wanted to focus on this class of upregulated microRNAs, which we validated by PCR, and we were able to confirm that indeed these microRNAs are overexpressed in physiological EBs compared to overloaded. We then, before moving on to the pathway analysis, we then wanted to cross-reference the predicted targets of these two microRNAs, which from the pathways analysis on MIRWalk were more than 2,600. To narrow down our analysis, we cross-checked the expression of these genes with a sequencing that we performed, a bulk sequencing that we performed on cardiomyocytes isolated from the slices. And what we found is that there are 6 targets in common. There are both predicted targets of our candidates microRNAs, and they are also overexpressed in overloaded cardiomyocytes compared to the physiological. These 6 genes that you can see here on the right underwent a pathway analysis, and from the biological function of reactome pathway, we saw that these microRNAs are highly associated with P10 stability, which is involved in apoptosis, and directly involved with the contractility of the muscle. So then we thought, what if physiologically these have a direct effect on the contractility of cardiomyocytes, which is our first outcome in the analysis? So what we did is we isolated cardiomyocytes from the slices by using, because we wanted to look at cardiomyocytes in isolation, so we used the iPSC-derived cardiomyocytes, which you can see here in this dish, and we quantified their contractility, again, after 48 hours of treatment with physiological EVs compared to non-treated cells. We found no significant difference in contractility in this case. So we also wanted to assess the calcium handling parameters that are also dysregulated in heart failure. So we moved on to also analyzing the calcium transient. You can see here this iPSC-derived cardiomyocytes, they beat upon 1 Hertz stimulation, and they contain already a calcium indicator within themselves, so they flash upon stimulation. And when we measured all the parameters of the amplitude of the calcium transient and the kinetics parameter, we did not notice any significant difference in the amplitude, although treatment with physiological EVs was slowing down the time to peak of this calcium transient. So in summary, we saw that physiological and overloaded slices exhibit different functional behavior after 48 hours of culture. EVs from rat overloaded slices have a significantly higher protein content compared to physiologically loaded slices. Overloaded EVs do not significantly affect the contractility of physiological slices, but physiological EVs do increase the active contraction of both physiological and overloaded slices with a minimum concentration of 10 to the 8 particles per slice. The effect is not mediated by a reduction in the cardiomyocyte size nor in development of fibrosis, but is likely due to a reduction in cell death and reduction in blood vessel rarefaction. MicroRNA's analysis revealed that the highly expressed miR378A3P and miR23A3P in rat physiological EVs are associated with not only a positive modulation of contractility, but also with cell death and angiogenesis. Physiological EVs then may affect the remodeling of overloaded myocardial slices, and understanding the mechanisms by which physiological EVs can mediate their effect in contractility might shed light on potential therapeutic candidates for the treatment of heart failure. And with this, I would like to thank both my supervisor, Professor Cesare Terracciano and Professor Costanza Emanueli at Imperial College, and the whole electrophysiology group. And of course, thanks the sponsor that funded my project. Thank you.
Thank you, Laura. What an impressive project. Scientific research are constantly looking for relevant and reproducible models looking for consistent findings. Well, now we have some minutes to respond to questions from the audience. Please, audience, feel free to send your questions to the chat box or even raise your hand and then you can open your mic. Meanwhile, I would like to ask a question if you agree, if you allow.
Sure.
This is most a reflexive question. When you say that you performed a microRNA microarray, are you talking about of the nanostring encounter, right?
Are you talking about what? Sorry?
Encounter, nanostring technology.
No, so for this microRNA array, I used a PCR kit from Qiagen, which was screening for the most highly conserved microRNA across both rat and human EVs. Just because I couldn't show all the data today, but I also work with human EVs, so I was looking at the most highly conserved microRNAs. So I focused on a set of 372 microRNAs to screen within, which are common between rat and human.
Fine. Okay, so the microarray that you're using can recognize the region of microRNA of humans and rats. That's it?
So mine was specific for rat because I used for the characterization, I used the rat extracellular vesicles. So it was rat microRNAs, but these microRNAs are also conserved in the human species. And in fact, I don't know if I can go back to my slides before. The microRNAs 23 is also expressed in human, so it has the prefix HSA.
That's the cause of my question, because I saw that you present some combined human and rat microRNA as potentially relevant for your study. And now I have a question. If you start the analysis evaluating rat samples, the presence of human microRNAs will be a factor to change our analytical protocols, or eventually it could be possible to have in sequence terms, not necessarily by species, but in terms of the sequence of some human microRNAs actually known for us, but no described for the rat microRNA group, and eventually present in vesicles.
So if I understood your question well, because I focused only on rat extracellular vesicles, but of course these microRNAs are also expressed in the human, I do not know if I analyze my human EVs with the same array if I find the same upregulation in microRNAs. I don't want to say yes, because I did notice when I was performing the analysis and the quantification of my human EVs, which I limited to just a basic characterization, I did find a significant increase in the release of failing EVs compared to the physiological from donor hearts, which I did not see in the rat. So potentially these two EVs from human and rat, although the contractile parameters do resemble human behavior, I don't know if the EVs have the same profile. I will have to investigate more on that. And is on my next step of the project, actually, because I wanted to expand on the human studies.
All us need to continue researching it. But okay, that's it. We can discuss more later. Thank you, Laura.
Thank you very much.
So for continuing our event, I will pass the mic to Atama. Another